Fact of the day.
Fact of the day.
Hello, hi, and welcome to Fact of the Day with me, Mark Lotsu. Monkey clones are humans next. So it's happened, the first known birth of primates using the same cloning method that was used to produce Dolly the sheep. This cloning method has also been used for about 23 other species as well. So this has been used to produce two little ickle baby macaque monkeys. They look so cute. And this is done in Zonghua, China. And funnily enough, one of them is named Zhong Zhong and the other one is named Hua Hua. I believe my pronunciation is correct. So researchers say it allowed their genes to be compared with genes from other monkeys that have had their genes tinkered with in order to do research into human therapeutic processes, drug screening, and mechanisms of disease. So what does this all mean for human cloning? So in 2013 and 2014, skin cells were taken and fused with an unfertilised enucleated egg cell. That means you have the nucleus out of the egg cell and you're basically putting the nucleus from the skin cell into the egg cell, and then with virus and electric shock to jumpstart it, it starts to develop into an embryo. Now for this process you can use fetal skin cells or you can use adult skin cells. Fetal skin cells are much more effective at working than adult skin cells are. But embryos produced in this way aren't allowed to progress past the 14-day stage. Cloning of humans is illegal in a lot of countries, but it's used for a process called therapeutic cloning where you can then look at embryo development and disease, and then they're obviously aborted when you get to the 14-day stage. So before you start freaking out about human clones being everywhere, like I say, it's illegal in a lot of countries, and also in the ones where it's not legal, it's not just a case of bish bash bosh, there you go, there's a human clone. It's a lot more complicated, So the two macaque monkeys were the only live births out of 79 embryos in 21 surrogate mothers using the fetal skin cell method. When it was attempted with the adult skin cell method, 181 embryos were implanted into 42 surrogate mothers. It produced just two baby monkeys, but they died soon after, one from developmental abnormalities. So to produce two baby monkeys, over 250 embryos were used in over 60 surrogate mothers. Now the maths of that is just ridiculous, and it's ludicrous to think that cloning is going to be a simple and easy process for us to be able to do. It doesn't work properly in other species, let alone humans or even the great apes. And also because it's in these little monkeys as opposed to, for example, sheep, in terms of being able to relate to it, it must have been quite tough for them. It's difficult, I think, to step back from this level of experimentation on monkeys. Human cloning is still a long way off, and even to think about or to begin to try to understand how to clone the great apes, that's still in the far, far distant future. Personally, I didn't think I'd feel this uneasy about human cloning, but it's still a long, long way off. That is, monkey clones are humans next. Thanks for tuning in. Love you, bye. Love you, bye. Love you, bye.